Good morning, good day. I don't know what time it is wherever you are. To you all, thank you for once again joining us for our community talks. Before we start, I would very much like to ask everyone who is not a speaker and attending this community talks to turn off your camera, as the internet connection is not always that great. If everyone turns off their cameras, this will make the internet connection a bit better. All right, so I will start with introducing today's speakers. So today's subject is mycotoxins, an insidious problem for human health in northern Uganda. We are very happy to be introducing speakers from both Gulu University in Uganda, as well as Ghent University in Belgium. We have Professor Geert Haesaert, we have Professor Richard Echodu, we have Dr. Godfrey Wokorach, who will be presenting. We are very happy to have them here today. As I read in the project file that we have here for us, it's also in collaboration with Muni University, if I'm correct. So yes, I think all that's left to do is to thank you for coming. If you have any questions, please put them in the chat. We will be taking note of them, and at the end of the presentation, we will go over all of the questions. So all that's left for me to do is to give the floor to our lovely speakers. So thank you, everyone.
Yeah, thank you, Larissa, and welcome to all of you. We are very happy that we can present results of two FLIR projects. We started 6 years ago, and today we are very proud that we can present the very nice results that we have obtained. So I will give a small introduction about mycotoxins and the source of mycotoxins. So we go to the next slide. Thank you. So what are mycotoxins? Mycotoxins are what we call secondary metabolites of fungal species. And it's normal that a fungal species produce secondary metabolites, but in some cases, these secondary metabolites are toxic for us, but also for animals. And the secondary metabolites or the mycotoxins are produced during infection process, but also further on during the colonization of the substrate. And that can be living plants, but also stored food or feed. When we look to the fungal species that can produce mycotoxins, and this fungal species we call toxigenic fungi, there is a broad assortment that can do that. And they are present in different environments, not only in the field, but also storage places, but even in silos we can find them. So different environments, in different environments we can find them back. They produce a wide variety of mycotoxins, and that means a wide variety of mycotoxins means also a large diversity in chemical structures, but also large diversity when we look to the toxicity level. Detection of mycotoxins is still a challenge, a big challenge, not only because of the different chemical structures, but also we must detect them in different matrices. And moreover, when we put a mycotoxin in a living organism, it starts to modify this structure. So, and then we end with what we call masked mycotoxins, modified mycotoxins, and also this needs specific analytical tools to detect them. When we look to the toxigenic fungal species, there is a big biodiversity between species, but also inside species. And diversity means that they can produce different mycotoxins, even in one species, different genotypes are able to produce different mycotoxins. When we focus on the most important genera that produce mycotoxins, so we end, of course, we must look to Fusarium species. A lot of Fusarium species are able to produce mycotoxins. I will not go into detail, but Fusarium species produce, for example, deoxynivalenol, nivalenol, zearalenol, T2, H-T2, fumonisins. These are the most important and also the most toxic one. The other species, group of species that are very famous for mycotoxin production are, of course, Aspergillus species. Also a lot of species belonging to this genera. And yeah, they produce especially aflatoxins, but also fumonisins. Some of them produce fumonisins and orgatoxin A. And the last group is penicillium species, penicillium species. Also, a lot of species can produce a big diversity in mycotoxins. And these are patholine, roquefortine C, of course, orgatoxin A, citrulline. These are the most important groups, but yeah, be aware that also Alternaria species, Claviceps species, Bisoclami species are able to produce mycotoxins in our food and feed chains. So I will now give the floor to my colleagues of Uganda, and I will present the results that we have obtained. Godfrey or Richard, the floor is yours.
Yes, welcome for the presentations. As Professor Geert has put across, mycotoxins are produced by diverse species of fungi that normally infect crops in the field, and the contamination level continues during storage. So normally people get exposed to mycotoxin unknowingly, because it is at times it is very difficult to observe with naked eye whether the food is contaminated. So when human beings get exposed to mycotoxins, it affects diverse organs, starting from the liver, kidney, reproductive systems. Some of the mycotoxins can cause damage in the DNA, and as a result, brings about mutations. So it's really the toxins that normally affects almost every organs within the systems. So for example, yes, for example, when we look at the impact of aflatoxin, aflatoxins, it is known to induce liver cancer when we are exposed to it for prolonged time. And the incidence of liver cancer that is induced by aflatoxin exposure is really high in Africa compared to other parts of Africa, as is highlighted in this pie chart, and followed by South Asia and Western Pacific. Aflatoxins also result into fatality when the exposure is very high, and periodic fatality has been reported within the East Africa since 1980s up to now, and each cases of aflatoxin poisoning, acute poisoning, results into a fatality rate between 25 to around 39, which is considered to be the highest worldwide, which highlight the the severity of aflatoxins and also mycotoxin problems within sub-Saharan Africa. So when we look at the level of aflatoxin exposure in humans, in Uganda, it is estimated that between 90 to 98% of people, when randomly samples are exposed to, they have at least aflatoxin biomarkers in their serums. For example, in the study done in northern Uganda by Leo et al. 2019 and 2020 indicates that the level of aflatoxin, especially in pregnant women, I mean the proportions of pregnant women that have a detectable level of aflatoxins in their serum is around 98%, which really indicate that nearly everybody in northern Uganda are frequently exposed to aflatoxins, and indicates also that the food that are mostly eaten within this area are contaminated with aflatoxin. So aflatoxins has both health effect and also economic effect. As highlighted here, the economic effect is that it normally results into what is called non-tariff trade barrier of agricultural products. Previously, Uganda and Tanzania had serious problems with Kenya due to high contamination of maize with aflatoxins, where Kenya banned Uganda maize and Tanzania maize from their markets. This ban normally causes serious economic problem to the farmers and also to the country, because it is estimated that at least Uganda loses about $577 million due to aflatoxin-induced trade barrier, and also like the neighboring country, Tanzania, loses about $332 million annually due to aflatoxin-induced trade restrictions on agricultural products. While within Africa, Partnership for Aflatoxin Control in Africa estimates that Africa normally loses around 670 million due to aflatoxin restrictions of agricultural products. Therefore, really, mycotoxins is a very serious problem that affects the health and so affects the trade. Therefore, when we look at the United Nations Sustainable Development Goals, number 1, 2, 3, aflatoxins really contribute to the greatest for like country in Africa or Sub-Saharan to attain to meet those goals of hunger reductions, poverty reductions, and also good health, given the health burdens and also the economic burden imposed by mycotoxins. Normally, contaminations with toxins are as a result of poor agricultural practices that relate to the method how crops are managed in the fields, and also how the food, when harvested, are managed at home, what we call the pre-harvest management and the post-harvest management. So, some of the post-harvest management that are key in aflatoxin contaminations or mycotoxin contaminations are drying methods, how the grains are dried, whether on a clean surface or on ground. Grain storage techniques that are critical in mycotoxin contaminations. And so this highlights some of the challenges of storage problems we have especially in northern Uganda, where farmers are really not having some of the best storage structure that can minimize post-harvest contaminations of food with aflatoxin or generally mycotoxins. So we conducted this study in northern Uganda. Uganda is one of the countries within East Africa. It is normally bordered by Kenya to the east and to the south by Tanzania and Rwanda, to the north by South Sudan, and to the east by Democratic Republic of Congo. So northern Uganda is located to the northern part of Uganda, and it is the part of Uganda that borders South Sudan. So there is a actually frequent interface between the people of Uganda and people of South Sudan. So the results indicate that majority of food that are really produced in Uganda, in northern part of Uganda, are contaminated with mycotoxins. For example, we observe that around 80% of sorghum is contaminated with aflatoxin, and 79% of groundnuts are contaminated with aflatoxins. This really highlights the kind of finding that we have observed that indicate that around 98% of people in northern Uganda, when samples have at least aflatoxin traces in their blood or in their urine. However, millet at the lowest level of contamination, and only around 21% of the millet that were sampled were contaminated with aflatoxin. Other mycotoxins like fumigicin was also frequently detected within the different food in northern Uganda. For example, 93% of sorghum within northern Uganda were contaminated with fumigicin, as well as 74% of groundnuts were contaminated with fumigicin. 67% of maize were contaminated with fumigicin. This really highlights that the greatest proportions of food are actually contaminated with these toxic chemicals. While for ochratoxins, the contamination was really high in sorghums and groundnut. For dawn, the contamination was high in maize and sorghum. So most of the food were actually contaminated with multiple toxins. For example, around over 20% of the food were contaminated with all the 4 mycotoxins: aflatoxin, fumigicin, ochratoxin, and DON. So this indicates that most of the people are really exposed to multiple mycotoxins in single diet. And for example, aflatoxin, fumigicin, and ochratoxin are already known to be toxins that can cause cancer. And exposure to all these toxins in single diet repeatedly really presents a severe problem to people of northern Uganda, and also the people who really depends on these food products. We compared the level of contaminations and the regulatory standards, where we observed that 71% of groundnuts really do not meet the criteria for this food to enter, especially better markets like in the European Union, while around 76% of sorghums do not meet the requirement, the aflatoxin requirements for the European standard. Therefore, majority of groundnut and sorghums which are produced within northern Uganda cannot be exported to European markets, which now highlight the problems within the problems that farmers in northern Uganda really face in terms of their food products getting into better market, so that they can really get some income from their food. As well, other toxins like fumigation, ochratoxin, and DON also present really a big challenge in terms of the food quality within northern Uganda to meet the international requirements. For example, for fumigation, around 70% of sorghums do not meet the international standard, and 40% of maize do not meet the international standard. Similarly, with ochratoxins, where greater proportion of sorghum and maize do not meet sorghum and groundnut do not meet the international standard. Similar to DON, where maize and sorghum had the highest proportions of food that, of samples that were contaminated beyond the regulatory limit that supports its export to international markets. So when we look at the level of aflatoxin, the level of these toxins in food compared with what is really considered to be safe, and what is considered to be safe, and it is measured in terms of tolerable daily intake, which refers to the daily quantity of mycotoxin evaluated as safe for human on a long-term basis in experimental animals. For fumigation, 1 microgram per kilogram of body weight is considered the level to be safe for human consumption. However, when we look at the level of fumigation in sorghum and maize, it is 8-fold higher than what is considered to be safe. And in infant, it's around 40-fold higher than the tolerable daily intake in sorghum, and 30-fold higher than the tolerable daily intake in maize. This indicates that the amount of this toxin that we ingest in northern Uganda is far above the level which is considered to be safe. And this really highlights a very big safety concern that require immediate interventions. Similarly, for mycotoxin or ochratoxins, the level is high. The level exceeds the tolerable daily intake, especially for infant in sorghum, which still indicate a safety concern that require immediate interventions. For DON, the level in DON exceeds the tolerable daily intake, both in infant and in adults. And it is really high for infants compared to adults. So this really indicates that very big problems for infants. And there has been a repeated observations that highlight children, especially in these mycotoxin hotspot areas, are really having high level of stunting, and which are an indicator of exposure to this high level of mycotoxin. Therefore, for us to reverse this kind of trend, like high stunting in children and other mycotoxin related problems, we have to reduce the level of intake to be below the tolerable daily intake. But at the moment, this really highlights there's a big challenge with food that we eat in Uganda. For aflatoxin exposure, the risk of exposure is measured in terms of what we call margin of exposure. So if the margin of exposure is below the thresholds, it really highlights that the level of aflatoxin intake presents really a cancer risk. For aflatoxin, we should not really eat even the smallest quantity, because it is a group 1 cancer agent. So it is known to induce cancer in the liver, and therefore its exposure really have to be avoided at all costs. But the exposure level for aflatoxin, both in human, both in infant and adult, really is very worrisome for the food in northern Uganda. For both groundnut, sorghums, and maize. However, for millet, the intake level does not really present a significant problem. Which in this study, we can propose that millet can actually act as an alternative food to diversify for diversifications, for this kind of for diversifications, like for sorghums and groundnuts that are really having high contamination with all the toxin. So for this case, millet can present some alternative in terms of really minimizing exposure to high level of toxin. So we have seen that there is really a big challenge to aflatoxin and other mycotoxins. Aflatoxin being a group 1 carcinogen, its exposure really have to be avoided at all costs. And therefore, this project looked at some of the mechanisms of controlling aflatoxin. One of the best alternatives, and one of the best methods to control aflatoxin is what we call biocontrols. Biocontrols of aflatoxin rely on getting that one that does not produce aflatoxin to outcompete variants that really produce aflatoxin. In many countries, like USA, has been already produced a product called aflasafe, TZ01, which is it has been demonstrated to control aflatoxin by 80 to 90% in the field, as well as in Kenya. They have produced a product called aflasafe KE01, and it has achieved aflatoxin reduction by 70 to 80% in the field. This is what we think we can replicate the same technology in Uganda, so that we can also reduce the extent of contaminations of food in northern Uganda by this level, by 90% or even more, so that it can really expose agricultural products in northern Uganda to better markets and also improve the income level of these poor people in northern Uganda who are really recovering from 20 decades year-long war, so that they can improve on both the livelihood in terms of income and also in terms of basic necessities like better food, better schooling, and so on and so on. So our studies demonstrated that really there are some isolates that we can really use for control of aflatoxin in the field. For example, we can see that the isolates, when we subject them to co-inoculations with some of the highly aflatoxigenic aspergillas, they are able to really control aflatoxin production by over 95%. This really presents a very good opportunity for us in Uganda to target aflatoxin control in the field by over 90%. So in this case, we have already demonstrated in the laboratory. The next step is now to demonstrate whether some of these isolates can really perform in the field as observed in the laboratory. So if we get this significant reduction in aflatoxin level in the field, then this would be really a magic a magic biocontrol agents that can be really fast-tracked to control aflatoxin problems in northern Uganda. So, yeah.
Okay. Thank you very much, Dr. Godfrey Wokorach. Just because from this presentation, what we have seen clearly is that the magnitude of the problem of mycotoxin is a very, very serious problem in Uganda, given that if you randomly select women who are pregnant, and you take their blood, you find that their blood has over 98% contamination in it. That is kind of very, very serious. And from what that we eat here cannot meet the international. So the level of mycocontamination is quite extremely very, very high. So for the last 6 years, when we have been into these two interface of the projects for Virel, we started first of all by initiating the concept of building infrastructure. The best way to be able to tackle this problem is first of all, the region or the area which is affected or the country needs to be able to put infrastructure so that we can be able to address some of those problems. In northern Uganda, we didn't have any surveillance going on until when this project came in, we started doing this. And through the university and the government then we put our priorities that we needed to be able to, first of all, start with putting infrastructure and then build on that. So I want to appreciate very, very much in that it helps us to stock the lab. This is a Gulu University multifunctional lab. It's basically entirely, the entire structure is full of labs, but we have designated labs, specialized labs, basically, which I'll be able to share later on, which are handling mycotoxin work, as well as fungal work. So we have been able to establish this in northern Uganda. So for the colleagues within Africa, Professor Baye from Mountain of the Moon and many other colleagues globally in Europe and in Africa, we have the facility if you're interested in doing any work within the context of mycotoxin, that we have HPLC, PCRs that can be able to do that in one designated area. So this is able to give us opportunity to be able to monitor what is going on in the field so that we can be able to educate the community. As I said, that our major input has been in the area of strengthening the lab. When we started, we didn't have anything. Unfortunately, we did not put some of those photos here. It was basically a bare ground, an open room. But gradually, when we established the lab, the context was, how do you go in terms of developing analysis platform? We started with basic ELISA, but ELISA is a quantitative one. So we needed to go into the specific one. And then we established the HPLC, as well as the sequencing unit for genotyping of the different strains of the fungus that we get across. So at the university currently, we do have this infrastructure, which is well established, which has been put to be able to do that. The other second key thing, after you have looked at the infrastructure, is to integrate with the farmers. Totally what we found in the field is that all our farmers are not aware of mycotoxin contamination. So one key area which we feel maybe moving forward as a country, we need to put more effort into creation awareness in terms of from the field, how should the farmer be able to handle their fields so that we can be able to reduce contamination? And then during the storage, what is the farmer supposed to do? In this part of the country, what happened is that we had a prolonged war that disintegrated the community, and the concept of food preservation kind of went off. So bringing it back is very, very critical so that we can be able to reduce this level where we are seeing over 90% of contamination in the foods that are eaten. So engaging the community is one of the things that we feel should be able to be done. And through this project, we did a lot of community engagement within those specific areas that we worked on. But still, there is still need more. There is more that we could be able to do to engage the community, to educate them how they can be able to control the mycotoxin. So here you'll see that within the collaboration that we had the colleagues from Ghent University, Professor Haesaert, together with me, with Godfrey, Wokorach, and another entire team, being able, first of all, to understand the dynamics from the fields, the storage, and engaging with them to share some of the results that we do have. The other key area that we went into creating awareness was engaging the media. We did 2 or 3 different types of engagement through the national media, like the newspapers of the country, through the radio stations, through the different talk shows. But when this report of ours came and said that 90% of the food eaten in northern Uganda is very highly contaminated or with mycotoxin, it got into the attention of the area members of parliament, and it was taken up into the parliament, so that specifically the government of Uganda starts to acknowledge and appreciate the dignity or the level of the gravity of mycotoxin contamination in Uganda, and specifically in northern Uganda. Within this, the motion was presented in parliament so that, first of all, the government prioritizes to create awareness in the local communities in the country and specifically in northern Uganda, but also should be able to put resources that can be able to generate solutions towards this. So through this grant, we were able to do that, and we are very appreciative that even up to the level of the government, we are able to see that policies are being adapted so that the idea of addressing mycotoxin doesn't become a Gulu university, doesn't become a Ghent university, but it's a national problem that needs urgent attention. We did also engage the different stakeholders within the community. Basically, the focus was on the extension staff of the different areas where we are studying. Specifically, we looked at the extension, the different agricultural extension services. We also fairly went into the medical centers within the region and present some of these findings towards them, but also be able to come up with the outputs in terms of the booklet. The key thing in Uganda, like a country which is having so many small dialects, several different languages, so we are also able to produce materials that we can be able to share, and we shared with these people so that when they go back to their communities where they're leading the agricultural component of improving the farmer's household, they should also be able to emphasize the need of integrating mycotoxin contamination so that the farmers are able to adapt that. We want to really thank very, very much Virel for supporting for the 2 rounds that we have gotten. It has brought us this far. We started from nursing, but this is how far we have gone. We have been able to train 1 PhD student, which is Godfrey, who has just given a talk on this, but that is still minimum because we need more young people to enter into the field, get training so that they take the mantle to be able to steer the country to address this monster, which we call mycotoxin contamination that is very high in our food. So we want to thank very much Ghent University for being with us for all these years. All the colleagues at Ghent University will still welcome you for additional areas. Currently, the areas that we are thinking of, we should be able to go into the aspect of biocontrol, but also deep go into soil so that we can evaluate and see these isolates that we have in the lab. Can we be able to make sure that we can try them in the field, package them, and possibly go into the industrial scale so that this can be able to be given to the farmers for purposes of reducing mycotoxin contamination. I also want to thank Gulu University, without the management of Gulu University, who have been extremely very supportive in having these activities run, as well as the community in northern Uganda. With that, I thank you people very much for listening to us, and I look forward for fruitful discussion. Thank you very much. You're welcome.
Larissa, we can't hear you. And you're unmuted. That's strange.
We are not hearing you.
No. We cannot hear you, Larissa.
Larissa, we are still not able to hear you. Larissa? Maybe I need to end my...
I think she needs to log in again.
Take some of these comments. Okay, we hope Larissa can get on board. So we are going to look at some of the questions that have been posted. Okay, Larissa is coming back.
Yes, I will let her in again.
Yeah.
Can you hear me now?
Yes.
Okay, great.
Good.
Very sorry for the technical.
So we have a couple of questions that we'll take care of. I will go through the chat and then be able to go question by question. Yeah,
So,
there is a need. Yes.
I wanted to add that I think the first question was asked in, was it in English or in Dutch? There's one question. I feel like this one was in Dutch. It was someone here in the chat who are currently also involved in the project with mycotoxins in ginger, and they were wondering if this project has come to an end or if there are new projects up and running, because they really would like to collaborate.
Yeah, so unfortunately, this project has come to an end. So we are trying to make sure we can put in the next call. Possibly there are some future coming calls so that we could be able to continue with this. But the lab still keeps on running. You know, this is a long-term investment. The infrastructure for the mycotoxin analysis, like ELISA, HPLC, and the entire lab is working. We still have some overlapping reagents remaining. So in terms of doing the work, we are still able to, but in terms of this project, it has remained. So we'll take more efforts to be able to contact the ginger team and the lead project teams to see if there is anything that we can be able to help them with the activities. Thank you very much.
Okay, great. And I think the rest of the questions you can read yourself. They were in English, I think.
Okay, there is another one here. How was this research data presented in Tabla were collected? I mean, what was it collected through the surveys? Yeah, I will ask Godfrey to answer some part, but specifically, we picked 12 districts in northern Uganda that captures a population of close to around 3 million, 4 million people in the area that we did work. And it involved two aspects. One was to interface with the farmers to kind of understand the dynamics in these small farmers, how their farming system goes on, and integrate and see how they are managing their fields, how they're managing the entire process. And then at the end of the day, we did ask them if they can give us some foods, and then we entered into the household, picked a couple of foods according to the standard operating procedures for doing this. So we picked the samples and then we came to the lab, did analysis. But the most interesting part of this work was, it was community-based. So you do analysis, make sure that the analysis is done well, and then we go back with the results to the community and say, okay, last time we picked the food here, and this is the level of contamination we found. If you don't mind, can you improve on A, B, C, D? And then again, we pick additional samples from that same household so that we can see, is the awareness campaign that we have done, as it entered into the farmers. So that's what we did. There was collection of samples, but also there was addition of questionnaire and then monitoring for a long period of time. It was a 4-year project. Do you want to add something?
Yes, thank you. as Professor Richard has stated, we collected samples from farmers, particularly we focus on the staple foods that are consumed in northern Uganda. The study focused on the awareness and also understanding the levels of contaminations. So the food samples were collected and brought to the lab. From the lab, the extraction of mycotoxins were done based on the standard operating procedures. Then the 4 mycotoxins were detected with the platforms that have been developed here. And also the second phase of the study was to look at the different mycotoxigenic fungi that are responsible for the contamination of food with the different mycotoxins. Knowing the fungi will help in the targeted response of their control strategies. So like if we understand that the predominant, what produce aflatoxin is predominantly A flavas, not A parasiticus, then we can understand the methods of how we can develop the biocontrols that target the toxigenic A flavas. And also, those are the major areas that we focus on. Look at the level of contamination, farmers' involvement, and also look at the biocontrol strategies that can be adopted.
Okay, we'll look at other methods, other comments. Okay, maybe reflect on, there is one here, table, mycotoxin prevalence in grains like millet is not self-explanatory. Okay, so we need to go to the table, which is showing the millet and sorghum self-explanatory. So what is very useful from those tables? So maybe if you have one, okay, somewhere here. Yeah, I think, yes.
Yeah, when you look at the presentations of the contaminations in terms of proportions of foods that were contaminated, these figures tend to highlight the level of the proportions of foods with different mycotoxin contamination. For example, we see that aflatoxin is predominantly found in sorghums, where over 80% of sorghums were contaminated with aflatoxin, and equally 79% of groundnuts were contaminated with aflatoxins. So this really highlights that nearly all the sorghum samples, sorghum food that are produced in this region are contaminated with aflatoxin. The same applied to fumonisin, where up to 93% of the samples were found to be contaminated with fumonisin. So the table, the figures try to highlight the proportions of foods that were really having contamination with the different toxins, and also try to relate with the findings of Lua et al. That indicate that over 98% of people in northern Uganda, when sampled randomly, you are able to detect aflatoxins in their serums.
Okay, thank you very much. So basically, what we see is that the tables and the figures basically show the proportion, and we are seeing that over 90% of the foods, like for example, fumonisin is contaminated, is having high contamination in the foods in sorghum specifically. The other second question is on the insight into community and the implication of indigenous knowledge versus the scientific knowledge. Yeah, thank you very much, Ben Dendela, for this question. We did kind of initiate community engagement. And I think we also want to thank Bilal that in future, we would expect that each and every project should be able to have a component of community engagement so that we can be able to see the knowledge that the farmers used to have. So when we conducted a workshop here in Gulu, where we called the different farmers and the agricultural officers, we did kind of get an insight, but we did not go deep into each of these communities to try to see some of the ways they are able to cope up with the situation. But what came out from that engagement is that previously, this community used to do adequate preparation of foods before they are stored. For example, they keep their foods into these food stores into the granary, but before that, they do process it. But because of the breakdown, as we said, this part of the country was kind of marched into war for close to 24 years until 2014, that's when things a little bit normalized. 2009, that's when things a bit normalized. 2007, things started normalizing a bit here. But because of that, there was that breakdown in the community, how they used to do community kind of engagement in terms of preserving food, in terms of processing them from the field, and a couple of other things. But what was also very, very clear, what these communities have adapted to, they do sow these seedlings which are infected. What was a bit annoying, or which was very unfortunate, is that they do sow these bad seeds, and then they use it for brewing alcohol. So that basically means the intake, much as we saw it in the food, into the beer consumption that people do take locally also, that's one of the areas that we see. So areas of pre and post-harvest management is kind of still lacking. So we need to strengthen that. And then the idea about food processing in terms of using the granary, they cannot use the granaries now because of insecurity. If you put your things in a granary, somebody is going to steal it. So I think the focal point here also, how can we improve on the post-harvest handling in terms of storage of food, handling it in the compound before it's stored. So we call upon different stakeholders. If you know where we can get these sacks, these good bags for keeping long storage of whatever, if possible, we can be able to get contact with some of the suppliers, private sector, which is dealing directly with this. We could encourage our farmers where to get some of this, but also we encourage our farmers to do what we call getting seed improvement. So those are the areas we feel we can be able to share and then compare with the knowledge of science so that we can be able to do that. There's something on, another question is on production of stable foods through biotechnology. Yeah, the country as a country, Uganda, we are doing breeding through the National Agricultural Research Organization, which are mandated to breed for new varieties that are resistant, and they are doing that. But also the government encourages most of these private sectors that are dealing directly with the seed distribution, that they improve, they bring down the improved seeds. From our study, what we found is that still there is low intake of the uptake of the improved varieties of seedlings in the community. Because of the indigenous knowledge, they still feel all the seeds needs to be retained. So I think, again, it goes to what we say, that can we improve the aspect of awareness so that there is an uptake in terms of the biotechnology, the improvement of the seeds into the farmers so that they can be able to use the seeds that can be able to protect or be able to be resistant to this infection. On the aspect of biotechnology, the government still doesn't have, we still don't have a law into place where we could be able to do direct application of GMOs, where we could be able to produce resistant things using the GM approach, genetic biotechnology. So there is a bill in Parliament which still needs to come into law, so that, but much work is currently being done in the lab to try some of the different genes which are very resistant, which we know can work very well. Until the law comes in, then we can be able to put this into the field. Do you want to add anything on that?
Yes, thank you once again. As pertains the biotechnology aspect, we have seen that one of the critical factors that predispose plants to fungal infection is the infestation by insect pests like fall armyworms. And studies have demonstrated that BT maize are really resistant to destruction by pests, and in a way, they have relatively low level of contaminations with mycotoxins. In Kenya, recently, they have already okayed the use of like GM product, which in the near future, we may see a real impact in terms of aflatoxin or mycotoxin reductions in that country. However, in Uganda, the bill is in parliament, which with time, maybe the country can also adopt the use of GM product in the control of mycotoxins. However, there are still also other approaches that we need to exploit that use microorganisms, like for example here the use of non-aflatoxigenic aspergillus flavus. That is also a biotechnology aspect of it. There are use of bacteria like lactobacillus, which we can still exploit, that can biodegrade some of these mycotoxins. So we can integrate multiple biotechnology approaches in reductions of mycotoxins.
Okay, there is another question, Professor Ribayo. Thank you very much, Professor, for the comment and reaching out to us. Prof, we are available. We need to build a team where we can be able to do the entire country survey and put into this data so that we let the leadership of the country be able to. So I think we'll be able to take this. Maybe you can drop us your email address where we could be able to expand in the future projects on the entire country. Rather than focusing Northern Uganda, we need to have data generated for the entire country for purposes of doing that. So you can drop us an email so that we can be able to see how we can work together for the benefit of the country. Yeah, there is an aspect of a lot of it in milk. Yeah, I think this is where there is the biggest problem. Studies have shown that mostly the byproduct end up into the milk, into our dairy because of the feeds that are given to the cows. So there's really a very, very big challenge. So I think maybe until we do some data and be able to present it, but other studies have already showed that dairy, egg production are some of the culprits, as well as the meat that we eat, where we see very, very high level of these mycotoxins in the food. Yeah, so they still need to have a lot of engagement. More research needs to be done. We need to localize and make sure that the health aspect is taken care of, and sensitize the community, the local leaders, so that the uptake, and currently we have already done that, but still more needs to be done so that the local community, our farmers, our leadership in the country at national level can take this very, very seriously. Now that the motion is already in parliament and is enacted as one of the policy going forward. Most patent and well-documented presentation is very interesting. Congratulations. Unfortunately, I have to leave now. Okay. But again, we want to thank Brel very much and our partners for being able to do this. So one thank Brel very much. I think, yes.
Yes, I want to bring to your attention that one of the critical methods that we can reduce exposure to mycotoxin is look at the diversification of diets, relying from predominantly staple crops that are highly susceptible to mycotoxins to less susceptible food types. For example, this study and the study done by Parker partner for aflatoxin control in Africa, highlights that sorghum is really susceptible to contaminations. To aflatoxin and other mycotoxins, but also generally have nation levels. So within this community, our study would suggest that people have to find a means of diversifying their food from predominantly sorghum to millet. Diversifications of food have helped in China, where they have reduced exposure level to over 80% when they switch from predominantly stable food crops that were highly contaminated to less susceptible food. Even in Tanzania, studies have shown that kids that have been fed on predominantly maize food have high level of exposure to aflatoxin compared to those with alternative food sources. Therefore, there is really need for us in Africa to try to expand and diversify our food to help minimize on the high level of exposure to mycotoxins. For the northern Uganda, we would believe that millet, as this result showed, can really provide an alternative for sorghums. That different studies have shown that it's really highly susceptible to contaminations with aflatoxins.
Okay. Any other?
I think those were maybe all of the questions. I can see that some people are sharing their emails in the chat, people who really would like to collaborate or have some nice ideas about mycotoxin and maybe some further research and collaboration. Of course, in our follow-up mail that we will be sending soon, we will include maybe ways to reach the speakers if you want to share maybe some nice ideas for new collaborations or projects. But I would very much like to thank all of you for being here today. And I, of course, very much like to thank all of the speakers. I feel like this is a project that can really change something for the communities in northern Uganda. And it's also nice to see that you were able to share this in the media and that you were able to share this in the Ugandan parliament. Really interesting subject, really interesting research. Of course, like I said, we will be sending a follow-up mail. Yes, I would very much like to also highlight that next month's community talk will be a special edition. We are going to be launching a community talk extra large, where we will be highlighting a closing of a long-term IUC project. So yeah, stay tuned for that. It's going to be a bit longer. It's going to be an extra large version. So you're all very welcome to join us next month. So once again, I would like to thank all of the speakers for joining us today. And yes, thank you for being here.
Yeah, thank you very much, Larissa, and the entire team at the Library Office, as well as all the community. I see a lot of colleagues from Ethiopia, Uganda, and many parts of the world. We appreciate, we'll take a bit of some time to copy all these email addresses so that we can start engaging with the different colleagues to see areas where we could be able to collaborate and be able to change the course of Africa in terms of addressing the challenge of mycotoxin. Again, I take opportunity to thank Professor Haesaert, Dr. Godfrey, Wokorach, and the entire team. Thank you very much for listening to us and for sharing and having a time with you together. Thank you.